Hey everybody, it's Miss Peachy from your WCA biology class, and doing a little lesson overview for Unit 3, Lesson 18, Disruption in the Cell Cycle. Just in case you're wondering what happened in the previous two lessons, I just decided that I didn't really need to do any kind of lesson supplemental for those. For lesson 16 and lesson 14. Again, they're talking more about modeling body systems and different body systems. The lessons are fairly short and they actually go through them pretty well. So just go ahead and read through those lessons on your own and make sure that you have the appropriate information that you need from them. So for this lesson, what we're really talking about is how can the cell cycle get disrupted? What happens if the cell does not function properly? Now, it says disruption in the cell cycle, but really, we've never talked about what the cell cycle actually is. So the first thing we need to do is kind of introduce the cell cycle and kind of talk about what a normal cell cycle is like. So I used a slightly different picture here than the one in the lesson. And this is the one I kind of put in your follow along. Whoops, that's the wrong sheet. In your follow along. Oh my gosh, it doesn't want me to grab the correct thing here. There we go. The follow along notes guide. So again, this is on my website. So go ahead and make sure you have this in front of you so that you're kind of following along with today's lesson. So the concept or what a cell cycle is, I like to think of it as kind of like the life cycle of a cell. So you can think of our life cycles and how we go through different phases of our life cycle. And we go from being born to dying, and there are many phases in between. The same is true for a cell as well. A cell has a point where it is formed, and then a point where it divides and forms a copy of itself, which is kind of the end of one cycle of the cell. So not all cells will go from birth to death per se, but they'll go from where they're formed to where they'll make a duplicate copy of themselves, which is shown here in yellow with the big letter M here. So we can start here with kind of the beginning of the cell cycle. So most cells aren't actively dividing in your body. And when a cell is not actively dividing, it is in this G0 resting state. It's just kind of waiting. So that's just true for a lot of cells. So even though in the lesson it kind of makes it seem like this is happening like every single day, it doesn't. It doesn't happen every single day. But when it does happen, when a cell needs to divide and make a copy of itself, it does take, depending on the cell, roughly the number of hours that are shown in the lesson for each phase of the cell cycle. Examples of maybe when a cell would need to divide. If a cell is damaged, it will need to replace itself, so it will divide and replace itself. If cells just age and eventually will need to make copies of themselves to maintain the integrity of your body. During growth periods, there's a lot of cell division that's happening. So when you're a child and an adolescent and you are growing, then you're going through lots of different cell division. It happens a lot. A lot of energy is needed for cell division. When you are an adult, you are done growing. You're not going to be dividing your cells as often or as frequently. But like, for example, your skin, it replaces itself about every month or so. So while the dead skin cells at the top, which when you have really dry skin, you can see those, kind of slough off, the new skin in the bottom of the epidermis are dividing and replacing the old ones. And your skin kind of just grows from the inside out and recycles itself about every month or so. So that's an example of fairly active cell division that occurs within your body. So starting at the end of mitosis here, we'll kind of start with the G1 phase. So the G1 phase is, during the G1 phase, you have the cell is growing at this point. Now it's preparing for cell division. So the end goal here is that you have a single cell that's going to eventually divide into two new cells, right? So we have our single cell here, and it has a nucleus that has all the DNA inside of it, right? And you got all these other like different organelles in here. I'm just going to draw them like different There's our endoplasmic reticulum and our Golgi bodies and our lysosomes and all sorts of stuff that's in here, right? And our mitochondria. So that's kind of what your cell looks like. And then you have the end goal of this is to be able to divide this up to make 2 new cells just like that. So the first thing that's got to happen is this thing's got to get bigger. So it grows. So it actually grows in size to about double the size that it was. At this point in the G1 phase, you still just have a nucleus with a single set of DNA, but you also double the number of organelles. So the organelles, you have to have way more organelles that form, right? So that you have enough organelles to go into the new cell. So the organelles are copied and replaced. So lots is happening in that G1 phase. So at the end of the G1 phase, you have this checkpoint right there. This is called the G1 checkpoint, and it's looking for any kind of making sure there's adequate nutrients, growth factors are present, if there's any damage to any of this new stuff that was just copied. We want to make sure that it's all kind of intact. And if everything passes muster, then it moves on to the S phase. If not, things can be fixed. There are enzymes that actually do go about and kind of repairing damage, but if damage is not, if it's irreparable or it's something that's going to cause, major dysfunction in the cell, then there's actually pre-programmed cell death that can occur, cell suicide, if you will. That's called apoptosis. And apoptosis, the cell will basically self-destruct at that point because it's just better to get rid of it before those mistakes kind of multiply later when that cell eventually divides itself, and then those mistakes get passed on. We don't want that to happen. So that's our checkpoint. Once it passes the checkpoint, it enters into the S phase. And in the S phase, now we got to double the amount of DNA that's found inside the nucleus here. So here in the nucleus, we only have a single set of chromosomes. So in the humans, that's 23 pairs or 46 chromosomes. So at this point, the 23 pairs of chromosomes will make copies of themselves, and you end up with 46 pairs or 92 chromosomes. So there's lots of chromosomes in there. So all of those chromosomes are going to be split up later on in the M phase so that you end up with 2 cells, each with 23 pairs of chromosomes at the very end. Okay, and then we have our second growth phase. And so the cell's got to continue to kind of get larger in size and kind of, again, making room for and making sure that it's ready for mitosis. There are certain proteins that have to be manufactured to be ready for mitosis. So there's lots of kind of preparatory work that happens. At the end of the G2 phase, there is another checkpoint to make sure the cell is the appropriate size, to make sure the DNA has all been replicated and everything's good to go. And again, you can have some fixes that happen. If, let's say, the DNA gets replicated and there's some of the code that's wrong. Some of the A, T's, C's, and G's are out of order, or there's some missing. Well, enzymes will come along and just make sure it puts those back into the right spot, or it'll splice out some of the things that don't belong and fix them. So DNA can be repaired and fixed. It doesn't automatically lead to apoptosis. Apoptosis is a last resort if the error is so major that it can't be revised and fixed. Once the G2 checkpoint is passed, then mitosis is entered into. During mitosis, the nuclear envelope that surrounds the nucleus dissolves, and all that DNA, which now we've got in humans 96 chromosomes, is divvied up. It splits apart. The organelles split. The whole cell gets kind of divided in 2 different segments with some DNA on this side, DNA on this side. So everybody gets like half of everything. And then eventually the cell will, the nuclear envelope will reform and you'll have 2 new nuclei. And then the cell itself, the cellular membrane, will reform and pinch off during cytokinesis and you have two cells. So one of the cells has kind of the old stuff, one of the cells has the new stuff, but you have two cells where once you only had one. And that's kind of the end of mitosis. That's the end result of the cell cycle. So again, this G0 phase, this resting phase, cells can spend a long time in there doing nothing and just kind of hanging out and being themselves and doing their normal cellular functions, what they need to do. But the actual process of the cell cycle is over and done with within about a day or so, plus or minus, depending on which cell we're talking about. So let's see here. So as we mentioned, we talked about apoptosis. This is the cell may self-destruct if, again, the mistakes are too big. Apoptosis destroys damaged, old, and unnecessary cells. It also, in the situation where your cell becomes damaged, you're not going to want that damaged cell to make a copy of itself. Instead, that cell will be destroyed through apoptosis, and a different cell that's healthy will divide to kind of replace the old cell. So the damaged, dying cells don't divide. Like the cell, I think your skin is a great example of this, because in your skin you've got like your epidermis, and there's multiple layers of your epidermis, and at the very bottom of your epidermis you've got these cells that divide and form new cells. At the very top of the epidermis are these really old and damaged cells that die. These guys aren't the ones that are going to undergo mitosis. It's the bottom ones that undergo mitosis, and they divide, make copies of themselves, and kind of get pushed upwards into kind of, the higher level. So there's different layers of cells here, multiple layers within your skin. So what happens when things go wrong? When you have the cell cycle that doesn't function correctly. Well, what happens is sometimes that DNA code that gets copied in the S phase of the cell cycle has errors in it. We've talked about those errors before. You've got point mutations that can result in substitutions or deletions or insertions that can result in frame shift mutations. Those we talked about previously. And one of the things that can result from this is cancer. And cancer is an overgrowth of cells with improperly or mutated DNA. Cancer is uncontrolled cell division. So mitosis just keeps happening over and over again, and you end up with a tumor in this situation. So a cancerous tumor is when the cells just don't know when to stop dividing. There's mechanism that tells the cell to stop the cell cycle is gone, and it just keeps copying. And oftentimes those cells are mutated, and so what's being copied is a mutated version of the original as well. So that's what cancer is. It is caused by mutations in the DNA that results in uncontrolled cell division. All right, so we've talked about the cell cycle. We've talked about checkpoints in the cell cycle. We've talked about apoptosis, cancer as a result of DNA mutations today. That's going to be it for this one, ladies and gentlemen. So let me know if you have any questions, and if not, I will see you in the next.